All right, we are going to start our chapter on genetics, and we're going to cover all the way through section 5.3, which is, we're going to do 5.3, which is non-Mendelian inheritance. This is your chapter on genetics. Please pay attention to this presentation. There are going to be some questions scattered throughout, so that I know you took the time to listen to this, and you're going to have to listen to it in class. So let's get started with genetics. I'm going to turn back in my book while I sit here so I can pull from the book if needed. So genetics starts with Gregor Mendel and simple inheritance. Gregor Mendel is known as the father of genetics with his famous pea plants, and we'll get into that in just a second. He began his life actually as Johann Mendel, and he was the son of an Austrian farmer. His father taught him about animal and plant reproduction, and he was completely fascinated by it. So that was his beginnings, and ultimately he joined the Augustinian monastery of St. Thomas and was given his new first name, and that's when he became Gregor Mendel. So, being a monk, he had a lot of time to investigate what intrigued him, and that's when he got into genetics, or figuring out about genetics. So, he used the garden pea with distinct traits, and something that he noticed, I'm going to draw on my presentation here, something that he noticed was that when he crossed a green seeded plant with a yellow seeded plant, that generation would give you, if you had two of those from the next generation that were both green, that they would always result in this consistent 3-to-1 ratio, where you would end up with 3 green seeded plants and 1 yellow seeded plant. And the way he explained this was that he proposed that each parent must have a factor that is passed down the generations that determine a specific trait. He called it elementin, but we call that a gene. He also proposed that the factor, or the gene, could be present in one of 2 forms, and we call those alleles. So alleles are different forms of the same gene. He not only looked at seed color, but he also investigated plant height, flower color, seed shape, and seed color. I'm sure he did many, many experiments. Let's talk about dominant and recessive alleles. So there are 2 alleles for seed color, 1 green and 1 yellow. The dominant allele is the one that expresses the trait whenever it's present. The recessive allele is the allele that expresses the trait only when the dominant allele is not present. Organisms have 2 alleles, 1 on each chromosome from the homologous pair. The alleles make up the genotype, so more than 1. 2 alleles per genotype, typically. Sometimes it can be multiple alleles. We'll get to that later. The genotype is the genetic composition of an organism, and it can be homozygous, meaning both of those alleles are the same. They're either both dominant or both recessive, or they can be heterozygous, where there's 1 dominant and 1 recessive. And then the phenotype is the physical appearance determined by the genes. And I know this is a lot to take in, but you'll start to see this in just a few minutes anyway. So I'm going to write here in purple, dominant. Those traits are typically written as a capital letter, and recessive alleles are written as a lowercase letter. So I've put capital R and lowercase r. So if they have 2, one on each chromosome from the homologous pair, then you would actually end up with the following: either capital R, capital R, capital R, lowercase r, or lowercase r, lowercase r. So when I say the alleles make up the genotype, this capital R, this capital R lowercase r, or this two lowercase r's would be an example of the genotype. So that's the genetic composition. Homozygous means both of the alleles are the same. So the ones that would be homozygous in this case would be the capital R, or the lowercase r. And then the heterozygous one would be the capital R, lowercase r. And I'll draw some arrows for you so you guys can see that. So then the phenotype, while the genotype tells us what's coded, the phenotype is the physical appearance. So if this R stands for seed color, and let's say green is dominant, then the capital R, and the capital R, lowercase r, would mean the plant has green seeds. But the phenotype is what is expressed. So the example here would be seed color. Okay, so dominant and recessive alleles. We kind of went over this before, and we used R, but let's go over it just in case. So each parent has two alleles for seed color. Let's do dark purple. The yellow seed allele is dominant. I think I said green in the last slide. So the yellow seed is dominant. That means if the allele for yellow is present, it will express. So a plant with two yellow seed dominant alleles is homozygous dominant. So that would be capital Y, capital Y, like shown here. A heterozygous genotype would mean that it has one of the dominant and one of the recessive. So capital Y dominant, lowercase y recessive. That's heterozygous. And then here's the different examples. Homozygous dominant be capital Y. Homozygous recessive, lowercase y. Heterozygous, capital Y, lowercase y. Now we have all this alphabet soup, but what does it all actually mean? First, let's talk about some rules that we kind of have to make. So the law of segregation is during gametogenesis, so when gametes are formed, each gamete only receives one allele for every gene. So you'll have the parental generations, those are the parents. In this case, it would be, changed to eraser by accident. In this case, it would be these guys. The first filial generation would be the first generation that comes from those parents. And the second filial generation would be these guys. And you see you see it again here, that 3-to-1 ratio that Mendel was talking about. So if we have a yellow parent, that's capital Y, and then you have a lowercase, or a green, which is homozygous recessive, all of the plants that result will have yellow seeds. Because remember, when the dominant one is present in this case, it's going to express. So all of these will have a heterozygous because there's two homozygous parents, one dominant, one recessive. So then that next generation, if you combine this here, you'll end up with two capital Y, lowercase y's, two lowercase y's, and one capital Y. That 3-to-1 ratio talking about what is expressed as the phenotype, which is what's seen. So how do we figure out the genotypes? We use this wonderful tool called the Punnett square, and you guys will have to really learn how to work on these. Learn how to work on these, practice these. You will see them on a test very soon. Okay, so you put the female gamete in the columns and the male gametes in the rows. So here's the females, and then here's the males. The male, just one, and one female. So how do we do this? You basically are going to combine the columns and the rows. So if you look at this column here, you have capital T here in the column and then capital T here in the row. So then this offspring will be capital T, capital T. And then if you look at this column and then this row, you'll have a lowercase t, which is always written second, just write the lowercase second, and then the capital T from the row. So capital T, lowercase t. And then same thing, do the next one. The column shows you capital T, and this one shows you lowercase t. And then this one is lowercase t, lowercase t. So if you look at these, all of these put together, you'll see that the probability of having a tall plant, this is what this one is looking at, a tall plant is 75% and 25% short. Why? Because 3 out of 4, which equals 75%, have the capital T, which when it's present will express. And then only 1 of the 4, 25%, has the lowercase t, lowercase t. Okay. So more complicated Punnett squares. What we've been working with is the monohybrid cross. So that is a single trait. So that's the single Punnett square that we looked at above. You guys will absolutely have to work and be able to do a dihybrid cross. So that's the results of male-female mating with regard to true traits. So really we're just combining Punnett squares at this point. So if you look at the little picture down here, capital T, capital P means tall purple flowers, and lowercase t, lowercase p is short white flowers. This one is written with the lowercase first, and for this I think that's okay. Make it a habit to try and write the capitals first because it makes it easier to see, but this will help keep it straight as far as going down the columns. And you see that it combines the same way. You're just combining what the column and the row give you for each letter. So that would be capital T, capital T, capital P, capital P. When you go down to the next level, capital T, capital T, capital P, lowercase p. The next one, capital T, lowercase t, capital P, capital P, capital T, capital, lowercase t, and then capital P, lowercase p. So then what you do is you see how many total options there end up being. That's a 4x4 square, so there are 16 total possibilities. And then you count how many are going to express tall with purple flowers, how many are going to express short with purple flowers, and then how many will express short with white flowers, and you calculate the percentages that way. You guys should be able to calculate percentages. If not, send me an email. So the law of independent assortment says that each allele for a trait moves into the gametes without influencing the other alleles for the other traits. And that's the only way that a Punnett square can work, because if we had alleles impacting each other, then the Punnett square would no longer work. Okay, so patterns of inheritance. I have here a picture of the greatest dog that ever lived in the history of the universe. His name was Winston, and he fits perfectly into this because we're going to talk about pedigrees in a second. And this is the day that I showed him, and he became a champion dog. And he is a German wire-haired pointer, or was a German wire-haired pointer, in case you've never seen one. They're the best dogs in the whole world. But back to biology. A pedigree is a record of ancestors for an individual or a family showing parent and offspring relationships across the generations. This can be helpful in understanding health issues that are inherited. It can also give you information for, how health issues, traits, you name it, it can be very helpful. So let's define a couple things. Autosomes Autosomes are chromosomes that do not determine the sex of an individual. And then sex chromosomes are 2 chromosomes that determine the sex of an individual. In humans, and typically most species, you have male, which ends up being XY, and then female, which is XX. Sometimes different letters are used, or they can be specific to to the species. So, for example, in birds, the males are ZZ and the females are ZW. Okay, so let's talk about patterns of inheritance. We're gonna talk about each one of these, but you're gonna learn autosomal dominant, autosomal recessive, and then a sex-linked disorder. And then within those, sex-linked dominant, sex-linked recessive, X-linked, Y-linked. So let's look at autosomal dominant. So autosomal dominant inheritance. Let's look at the symbols and the connections because they can be a little bit difficult. So males are shown here as squares and the females are circles. And most of the time it's males are squares, females are circles. Sorry guys, y'all are all squares. If the phenotype is present, meaning the trait or genetic disorder is present in that individual, meaning they have it, the circle or the square will be colored in. If it is absent, then that circle or square will not be colored in. So if we have autosomal dominance, it will be present. If it is present, it will be expressed. Remember, if it has a capital, I'm going to use R again. It doesn't matter if it's 2 capital R's or 1 capital R and a lowercase. If it's present, it will express. Individuals without the disorder will be homozygous recessive. So that would be, in this case, lowercase r, they would not have it, totally recessive. So if you look at this picture on the right side, we're looking at autosomal dominant inheritance. You have a female that does not have the disorder and a male that does have the disorder. And then they have 4 kids. Looks like 2 girls and 2 boys. Of their kids, you have to look at the lines that connect directly to the above. So those are the 4 kids. I just put a purple line. And then you see the 2 girls and then the 2 boys. So of their kids, 3 of their kids have the disorder. So then let's look at the pairings from there. So this daughter who has it marries this man who does not have it. And then you can see they have 4 children. And then 1 boy and 1 girl from these 4 kids have it. And then if you look on the other side, you have a male child affected with the disorder marries a female who is not affected and then has 4 kids. And there's a boy and a girl that each have it. So it doesn't matter. It affects, because it's on an autosome, it's not going to matter if it's male or female. It's going to affect basically equally, and you see that across the board. There are 2 females up here that have it in the children, but if you look down the line, in each case, the F2 generation has a male and a female on both those sides. So this is autosomal dominant. So if the phenotype is present, it's dominant. All right, so genetic disorders. We can look at it as, is it autosomal recessive? You can determine that is by asking a couple questions. Is there an unaffected couple that has an affected child. Is there a couple where both are affected, and do they have affected children? Does the condition appear equally in both male and females? So let's look at the picture on the right for autosomal recessive inheritance. You see here there is a male and a female. They get married and they have 1, 2, 3. They have 6 kids. The male is affected, and we're saying that this is autosomal recessive, so lowercase, meaning the only way that it's expressed is if a child ends up with this genotype. So we look down the line, and they have 6 kids. They have 4 kids. 1, 2, 3, 4, and only one of those children is affected and has it. But that child looks like they marry an affected female, and then every single one of their kids has it. So that must mean that they were lowercase r for this trait, lowercase r, and lowercase r, because the only thing that could happen for all 4 of those kids to have it would be for both of them to be autosomal recessive for that trait. Now, if we look at over here, we have this male marries a female, and neither of them have it, but they must both carry it. So that must mean that they are heterozygous, capital R, lowercase r. Why? Because remember, if it's autosomal, they must get 2 lowercase r's, and the only way that can happen is if they were heterozygous. And that's how this child ended up being affected. So that's autosomal recessive. So sex-linked inheritance. Why does it affect more? Well, first of all, a sex-linked disorder is a disorder caused by an allele found on a sex chromosome. The allele could be dominant or recessive, so some genetic disorders follow a sex-linked dominant pattern, and others follow a sex-linked recessive pattern. So you can divide them. Since X and Y chromosomes are different, you can also divide the sex-linked disorders into X-linked and Y-linked. They're a little bit confusing right now, but we have some examples to go through, and I think most of these end up being sex-linked with it on the X chromosome, which is a lot of times what you see because everybody gets an X chromosome, but not everyone gets a Y chromosome. Hold on just a second. I'm trying to find the page so I can kind of... Okay, let's talk about specifically, in this case, we're going to look at okay, so for now, I think we're just going to talk about, like I said, it's more complicated with the X and the Y, but we're going to discuss sex-linked recessive inheritance related to the X chromosome. And a lot of those are fairly common. Some of those are color blindness, which is a recessive disorder caused by abnormal alleles on the X chromosome. Males only have one X chromosome, so a male with normal color vision must have normal X chromosome. His Y chromosome doesn't matter since the relevant genes are not found on that chromosome to link to color blindness. A female, on the other hand, has 2 X chromosomes. Since color blindness is recessive, she would only need 1 normal X chromosome to have normal color vision. Both of her X chromosomes could be normal, but it only takes 1 normal X chromosome for her to have normal vision. So let's look at, this example comes straight from your book, for sex-linked recessive inheritance, meaning we got to have a lowercase, 2 lowercases, and we're looking at an allele on the X chromosome in this case. So you have up here a male and a female, they get married, they have, in this case, I think almost in all the cases, they have 4 kids. And then they end up having 1 affected daughter and 1 affected son. number 3, and number 7 in this picture. And then you look at this next group of 4, and you see none of them are affected. Meaning they may be carriers, but they're not affected because it must not be recessive. So none of these have the lowercase r. But on this side, you see the female is the one that's affected and expresses it. So, and then this one, hold on, I'm trying to think. I gotta think about this for just a second. Let me look at number 4. Person number 4. So if you look at parents 3 and 4, this woman is colorblind, and they're using colorblindness as the case. She has 2 recessive chromosomes. The man is not, so he has a normal one. All the female children get 1 X chromosome from their father and 1 from their mother. Since the father's X chromosome is normal, all the female children get 1 normal chromosome, so none of them are colorblind. However, the male children get their X chromosome from their mother, and both of hers are recessive. So that's why all the males here end up colorblind. Okay, so we've talked about these are very specific, and we call this Mendelian inheritance. And then your question that you need to answer, so that I know you watch this, is what is the name and breed of my dog from the picture, the pedigree picture? Okay, so non-Mendelian inheritance. What does that mean? So while what we've been talking about is things that end up with very specific traits and very specific outcomes, there are some traits that are not totally decided on one thing. For example, if you look at certain species of flowers, for like carnations, they can be red, white, or pink. The white and the red are the 2 alleles. Let's just say that. One would code for red, one would code for white. But you end up with pink. So you have this incomplete dominance. So the alleles blend together to produce the phenotype. And so you'll get somewhere in the range between the red and the white, in this case pink. This can also be called codominance. And then there is another exception to Mendelian inheritance. Evident is that there are multiple alleles that are responsible for producing the phenotype. For example, in blood type, it's the most familiar one we have. A cell has many proteins on the surface, and some of them act as antigens, which help other cells identify it, like red blood cells, and it becomes important when receiving donated blood. So you know about blood typing. There is type A, type B, type AB, and O, and they are all related to a single antigen produced by a single gene. In this gene, there are 2 alleles that are codominant. A and B are codominant. O is recessive. And then you also have to consider the Rh factor. It can be positive or negative. The factor is based on the second gene that follows simple Mendelian genetics. So the Rh positive is dominant and Rh negative is recessive. So blood type is usually reported with letters followed by a positive or a negative. If your genotype is AO plus minus, then you have an A allele, an O allele, and a plus. So you would, since A is dominant over O and plus is dominant over negative, you would be A positive. But yeah, so I highly recommend that you do the comprehension check about blood type and redo it if you got it wrong. Because I know for a fact your test has a question about blood type and what is expressed with percentages. So I want you to make sure you spend some extra time on that. Okay, so one last thing to talk about non-Mendelian inheritance is polygenic. This is multiple genes working together. For example, skin color. If you look on page 142 in figure 5.11, there's a hypothetical Punnett square for skin color. And this is assuming there were only 3 genes that were working together for skin color. But in actuality, there are many, many more genes that work together for skin color. And you can easily see how, even if it was just 3 genes, how many possibilities exist. And you can clearly see how Adam and Eve could have genotypes for many, many, many skin colors. And then skin color is also impacted by the environment, which is in the next section. And we'll talk about that when I get back. For your test, you must be able to do a monohybrid, so single trait, and a dihybrid cross, so 2 traits, like the tall purple flowers. And this one would be just one like that in a Punnett square. We will practice these. You have a few on the in-class assignment, and you guys now have the rest of class to work on the in-class assignment. If you finish it, remember, and you do your best effort, I will replace your lowest test grade with a 100. I'll see y'all next week.